Okay, we're now going to move on to hear about tar spot genetics from Addie Thompson at Michigan State. Thank you very much, Addie.
Great, thank you, and thanks for the opportunity to speak today. I'm excited to be here. A little bit about me. This is the background that you'll see when you look at sort of my biography. And I did do my undergraduate here at Iowa State, although I will mention I was in the College of LAS. So I put this here for two reasons. One is to say that during all this, I also had 2 kids, so if you want to talk to me about any of that or work-life balance, please come see me tonight at the diversity dinner, that'd be great. But the second reason is that a lot of plant breeders will come up and talk about how they grew up on a farm and they worked detasseling corn, or, I don't have any of that background. My family was not on a farm, and I came here from a very different route. And I say that to welcome those of you who are not from a farming background, but you find you have a place in plant breeding. That's great. The reason I'm here, I'm the product of a lot of different youth programs, and not even just one, but several. So I'm so excited to see that the MPGS is including high school students and youth and getting them involved in agriculture, because we definitely need that. So currently I'm at Michigan State. I do research and teaching in mostly maize, and I work on genetic mapping and genomic prediction, high throughput phenotyping, and innovating modeling and prediction for breeding. And I also have a passion for teaching, especially in transdisciplinary skill sets, mostly using hands-on, project-based experiential learning. And I also do teach the advanced plant breeding course the first semester. Francisco Gomez over there, he teaches the second semester. So we're fresh insight into the plant breeding at Michigan State. So this is kind of what my field program looks like, some field corn, sweet corn, and sorghum, other crops, which then looks something like this. So I don't have any students here this year, unfortunately, because they're all out in the nursery. Our field season is about a week delayed from you guys, so we're still kind of just coming off the peak. So they're still out there every day until 5 or so. So today I'm going to talk most about the critical role of germplasm in my program, and focusing on mostly tar spot, but I'll touch on quantitative genetics and phenomics and a little bit about the educational piece I mentioned. Now, the reason germplasm, obviously it's critical to any breeding program, but especially at Michigan State. The last person that was there that had a corn breeding program was Elmer Rossman. So he was there for 42 years, but you'll see he passed away in 1989. They had not hired a maize person since then. So instead of inheriting a rich plant breeding history and germplasm, I had nothing. I was starting from scratch. So of course, the first thing that I did was I contacted the MPGS. And I said, hey guys, I need some germplasm. And not knowing even where to start in Michigan, I thought that perhaps I could start with what had worked in Wisconsin. So I pulled out the Wisconsin Diversity Panel, and I put it in order in 2018 for probably close to 1,000 lines. So I noticed that you said the average was 22. I was well above that average. But they didn't even bat an eyelash. They're like, yeah, sure, no problem, which was amazing, so thank you. So I started in January of 2018, and I was able to hit the ground running with phenotyping and increases in the nursery that first summer, which was incredible. So that has been grown and phenotyped and increased in Michigan the last few years. But I also tried to pay it forward. So several people here and the maize community have benefited from that germplasm because I've shared with other groups as well. So we have kind of a little network going on. What you see here in the picture is actually a photo of the boxes that were sent to Nebraska. This picture was taken March of 2020. So you might remember where you were in March of 2020, but this was right before our lab shut down. We prioritized getting everything packaged and shipped out to all these different locations so that they would all have seed for their summers, even during the pandemic. And that has since resulted in some collaborations and interesting discoveries. So I wanted to take a moment. Dr. Kantar and I were discussing last night, Dr. Ron Phillips at the University of Minnesota, and I got to thinking about some of the things that I had learned from him and I had taken away from my time at Minnesota. And the main 2 ones were, first, to always be above reproach, but the second was about opportunity, and to always conduct your research in such a way that you will trust your data and you're poised to take advantage of any opportunities that arise. And his example of that was, let's say you collect data in an experiment and you get some strange result. Are you going to trust that that's a really exciting new discovery, or are you likely to think that that's an error because you did not conduct the experiment in such a way that you really trust what you're looking at? So I've taken that to heart, but also I think that applies to things like your general breeding program and to germplasm as well. In my case, I spent the first several months of my appointment talking to as many people as I could and finding new collaborators. And one of the people that I talked with was Marty Chilvers, who's the fields crops pathologist at Michigan State. And he said, we have this new disease. It's called tar spot, and it's going to be really important. It had been around in Central and South America for the last 100 years, but it was new to the upper Midwest as of 2015. So this is caused by a pathogen, Phylochoromatus. It's an ascomycete fungi. Obviously its host is maize. It is an obligate, and we cannot inoculate for it, so one of the challenges is you have to have a region that has high incidence. It's spread by wind and water, and it causes these raised, shiny black spots on the leaves that looks like flecks of tar. It thrives in cool, humid conditions, spreads quickly, and can cause up to 40% yield loss. And I'm showing here a picture with, you can see there's varying sizes of spots. The key is that you can't scratch them off. So if you scratch it off, that's just insect frass. But it can have these necrotic lesions we call fish eyes, or it could not. It covers the leaves pretty rapidly, reduces the photosynthetic capacity, and it can quickly break down the canopy. So it kind of pulls nutrients out of the stalks and increases the propensity of lodging. This is a drone aerial imagery taken by Marty's group where the whole field has really high levels of disease, but you see this area is especially very, very highly affected. And just a short time later, this is not what maize should look like in Michigan. That time of year it should not be, it should be green and happy. But you see this is all completely lodged. So it can really move very fast and take out large areas. So the opportunity that we had then, we had this diverse set of germplasm we'd already increased. We were all poised to work with it. And we decided we were going to screen for resistance. I also then contacted the repository again and I said, hey, I heard you have these cool gem lines. Can we screen those too? So they helped us select a set of 100 gem lines and 100 B gem lines that they thought would actually mature in Michigan, and they did, so thank you. And we took weekly ratings and assessed other phenotypic traits. So this is just an example of our rating panel that we used. And meanwhile, we started up a USDA-funded project that we called the Great Lakes Tar Spot Initiative. Give it a flashy name. And what I did is I paired plant pathologists and maize breeders at each of the regions around the Great Lakes. And then that gave us an area that we could use to screen germplasm at different locations. I'll point you to this resource. This website, you can go to track tar spot in any given year. So the gray is where it has been reported previously. The orangish color here is where it is currently positive for this season. And then yellow is probable, et cetera. So as you can see, it has been located throughout the upper Midwest there. And I think it's poised and ready that if there's a cool, wet season, it could become a problem. Obviously, in a year like this where we've got hot, dry weather, you won't hear much grumbling, especially in Iowa, but I think given the right year, it could be a big issue. So what have we learned in this project? First, I will say we found resistance, which is great. So these are two neighboring plots, and you can see this leaf is very wet, so it's not a lack of leaf moisture. So we did find some resistant varieties, and we've started introgressing those, which I'll get to in a minute. One of the first questions I get from breeders is, where is it coming from? What sort of germplasm pools have resistance? And the answer is you can find some level of resistance in many of the germplasm pools. So there's not like a major resistance gene per se, but I think there are susceptible and resistant varieties spread across many of the common germplasm pools. I'll put it that way. So we have done some GWAS analysis on our different locations of trials. Here's an example from one of the Michigan locations in 2020. And then the next question I will get often is about the gem lines. So this is a plot of the disease incidence by population and year. And you'll note that almost every plot gets some level, although not every plot. And the gem lines are a little bit slower to accumulate disease. But I add a caveat that it's a little bit confounded because we do have other diseases that are present. And the gem lines really a lot of them were not resistant to rust. So that was the big one, that because this is an obligate pathogen, it has to have green, healthy tissue. And so if you have a plot that gets decimated by rust, there's not a lot left to be hit with tar spot. So ongoing progress. We have obviously mapped resistance. We've identified some resistant varieties. Those have been backcrossed into some elite temperate field and sweet corn varieties. The sweet corn I'm not going to talk about, but we have some populations we're working with this summer for further screening. For the field corn side, we've been developing genetic populations for further study. So there are a number of DH lines, double haploid lines, thank you for that introduction on double haploids, with both B73 for the genetic side and then with LH244, and we're using those populations as test beds for their technologies. So one of my students works on image analysis and is hoping to create kind of like an app that you can take a picture of the leaf and it'll tell you the quantification of the tar spots. And then we're working with validation and further mapping of the resistance. One of the resources we used this past summer, 2021, was a stiff stock magic set of lines from Sean Kepler's group, so we grew that in 3 states, and weirdly found some resistance coming from B73. So maybe that's the reason some people have noted that there's some resistance in your program. So B73 we were using as a semi-susceptible variety to cross our resistance into. We took about 5 different resistance varieties and crossed those in, but I think we'll actually get some resistance from both parents. But those DH lines have been genotyped already and increased, and they're being tested in 3 states this summer, so I'm excited to get the results of those trials. The LH244 lines are being increased this summer, so that's why the nursery is so crazy, is because we have that extra set of hundreds of lines, but those will be tested next year. All useful material will be deposited into the repository for future use, so along with those phenotype and genotype information, so be looking for that. Meanwhile, we have just started up as our first USDA project winds down. The second one is starting to ramp up. So we called this the Tar Spot Initiative 2. We're looking more about the mechanisms and relating phenolic compound accumulation with tar spot resistance and using hyperspectral approaches in sort of 3 different scales: powdered tissue, leaf scans, and then drone imagery. And then doing some validation and testing of candidate resistance genes. And I mentioned this drone imagery, we get the visible near infrared and shortwave infrared for the spectral information, but it also has LiDAR built in. So LiDAR stands for light detection and ranging. You basically have lasers bouncing off of objects, coming back, and you get an idea of the shape of what you're looking at. So this is an example of a plot across 4 different dates, and you're seeing kind of the shape. And we think that that actually could be useful as well, because again, this causes degradation of the canopy and lodging, so maybe we can kind of characterize that using the LiDAR data. With the Phenomics piece and the tar spot project, we're hoping that we can improve the detection of onset and severity as a way to maybe identify disease before it's abundantly visible, which would enable more management options for farmers, because there are fungicides that work relatively well if you get the timing right. Okay, for other projects, we do participate in the Genomes to Fields project, which if you've not heard about, it's a collaboration of about 20 different groups across the US and into Canada now. We share germplasm and we collect data. Someone might be talking about that later today or tomorrow. We'll find out. Otherwise, you can ask me questions. We're interested in how we can model and predict into other environments. And in doing that, we're trying to sort of parameterize models for the different varieties. And so we have been painstakingly measuring thousands of plants, which is great, but it's also incredibly slow. So our thought process in Phenomics is, can we use some sensor technologies to measure traits and then model more complex traits to then get at these composite traits? So the goal is that we'd be looking at things that are more heritable, but then also using crop growth models to sort of combine these aspects into the outputs rather than going from genetics to yield directly. And just as sort of a thought experiment, I had put together what sensors we might use to get to each of these traits. And for some of these, we had experience and kind of thought, yeah, this sounds like it could be possible. But the one that we had not tried, because not a lot of people had used it yet, was the LiDAR. We did not know what all of these traits you could get. And so just as an experiment, preliminary data, if you will, we took our LiDAR data and used it directly in a neural net to try and predict plant traits. So this was a collaboration with Daniel Morris, who's in the computer vision group at Michigan State. And so he did some phenomic prediction on a number of different traits. So flowering time, heights, ear leaf number, total leaf number, and leaf area. And we found for a lot of traits that some temporal data is important. I won't go into great detail on this, but overall you can see that we're getting pretty good predictive accuracy. And we did compare this with genomic prediction, which I will say is an unfair comparison, because when you're looking at phenomics, you're looking at both genotype and the environment and what that plot is doing, but it's still meaningful for breeders, I think, because it's a real-world scenario of how are you going to allocate your resources to make the most impact on your selection. So we did compare genomic and phenotypic prediction and noted that our LiDAR prediction approaches were actually quite good in a lot of cases for most traits. I'm going over some of this pretty rapidly because I'm on a time crunch, but please feel free to talk to me later if you have questions about anything or what to discuss. So other projects that we're working in, I have one student that's working on multi-objective genomic mating optimization for breeding programs. So like I said, a lot of the work that we do isn't necessarily breeding per se, but we do a lot of breeding-related research. So there's a number of other projects that I could happily talk about if you're interested. But I want to get to this last bit, and that's on education. So Michigan State has a program that's called IMPACTS, which stands for Interfacing Computational and Plant Sciences. This was an NSF-funded research training grant where we bring graduate students in from both the plant science and the computer science side. I have some examples here in the front. You can talk to McKenna. But I teach the second course in this series, which is called Frontiers in Computational and Plant Sciences. And it's project-oriented and team-based and really emphasizes interdisciplinary communication skills. So I joke that I don't teach them the science, I teach them how to work with each other in order to accomplish important questions. And I used to put an XKCD comic here about learning what's hard in computer science, but instead I've switched to this quote by Temple Grandin, which is that the world needs different kinds of minds to work together. And finding a common language with diverse collaborators can take a lot of time, but it's really beneficial. In this case, I'll give you a couple quick highlights. This is from last year's spring course. It was a partnership with the Syngenta sweet corn breeder, and she provided these images of sweet corn ears on trays, and one of the questions they were interested in was blank fill. So how often do you get kernels versus the ear showing? And so the students who had no expertise in image analysis whatsoever worked together they learned from the breeder, they learned from an image expert at Michigan State, and were able to put together, this is just one of the group projects, a pipeline where they were calculating total area of the ears and area of the kernels and then taking the difference. We've since partnered with a number of other groups, including public and private. So we actually drove the EarthSense TerraSentia rover that you maybe saw on Monday out at Corteva. We drove that through a Weaver popcorn trial out in Ohio. And this was one of the groups this past spring that was looking at sort of canopy architecture in the field from LiDAR. And then another group was looking at hyperspectral analysis of potato plots. These are where's Dave? These are Dave Douch's potato plots. And so they were looking at characterization of disease resistance using spectral data. But the reason I show you these two projects in particular is I want to show you where they started. So these are very, very short modules. So we do 3 of these in a semester. So each group has a very limited time that they can learn how to work with that data set. And they do kind of a project proposal and then an update and a final report. So at their first pass, this was the LiDAR group. This is all the corn. This is the sky where it's hitting the infinite. So they came back. This is not plants. So we had a lot of iterations that we had to go through. But I think going from this to some interesting results in the matter of a few weeks is really impressive. And that's the kind of education that I think really benefits graduate students for their future. So with that, I'll list acknowledgements. I spoke about some of the blue names today, acknowledge some of the funding sources, and I want to say thank you to you guys and to my team who's out in the field. But also, if you have questions, come find me, or here's some contact information that you can reach me. Thanks.